Or maybe I never started. I thought I had this going recording. But the bottom of page 230, where else does electron transport take place that we have talked about? Well, what preceded talking about respiration? Not in animals, but rather what instead? They're green in plants. That's right. Okay, so this electron transport came from a certain molecule being split, meaning that, okay, this is from the organic class that I want to leave up here, okay? So these hydrogen ions that are positively charged are pushing this electron down a chain so it can be released into the atmosphere, the electron transport chain. Okay, so on page 230, and we're talking about electron transport now. In aerobic respiration, electron transport is the final step in the breakdown of glucose. It also is the point at which most of the ATP is produced. High energy electrons and hydrogen ions from NADH. So notice it's talking about ions, just like it would here with photosynthesis. It's the hydrogen ions that push those electrons down this chain. Okay? So this NADH and FADH, okay, so they're produced, yes, we understand that, in the Krebs cycle, are used to convert ADP to ATP. Now that's something that is not new. We've talked about that probably 3, 4, 5 times, talking about how and why ATP formation is so significant. Okay, the next page. Okay. As shown in figure 14, electrons move along the mitochondrial membrane from one protein to another. Okay, and they release these electrons. The energy carriers are converted to these molecules. We're not too concerned about that. The hydrogen ions are pumped into the mitochondrial matrix across the inner mitochondrial membrane. Hydrogen ions then diffuse down their concentration gradient back across the membrane and into the matrix through ATP synthase. Okay? Did I mispronounce that this whole time? I thought it was synthetase. Oh, it is ATP synthase. Okay. I've been mispronouncing it this whole time. Okay. So this ATP synthase molecules in chemoosmosis. Okay. Electron transport and chemiosmosis in cellular respiration are similar to those processes in photosynthesis. Okay? Oxygen is the final electron acceptor in the electron transport system in cellular respiration. Protons and electrons are transported to are transferred to oxygen to form water. Overall, electron transport produces 32 ATP. Each NADH molecule produces 3 ATP, and each group of the 3 FADH2 molecules produces 2 ATP. In eukaryotes, one molecule of glucose yields 36 ATP under ideal conditions. Okay? So, through this entire process, how many ATP do you get? And it's not this answer. We're going to go with 36. Okay? I wasn't quite sure what the author had said, but we're going to say 36. So in this entire process of respiration, how many ATP do you produce from one molecule of glucose? Well, if we're producing 36 over here, what do we have left from glycolysis? What do we have left over here? So that must mean you're producing 38 ATP molecules, with every molecule of glucose that you have, which is quite an astounding feat to when you think about, okay, well, where did this glucose come from today? Well, it came from part of your chicken tenders, if you had that today. But we think, well, if it came from that, that's probably protein. Well, that's true, but what surrounded the chicken tenders? Say that again. And that breading is made of starch. Starch is made up of a whole bunch of glucose molecules. So that's going to be one of the sources that's in our diet where we get these glucose molecules. Okay. But we're not concerned about prokaryotic cellular respiration. Does anyone think they might know why that is? Why not consider prokaryotic? Because what type of organisms is that if you're talking about prokaryotic? Bacteria. Right. So it's just single-celled organisms. We're not concerned about that at all. Okay. So at the bottom of page 231, okay, it's talking about anaerobic respiration, which that's going into different types of fermentations, and alcohol fermentation is a type, lactic acid fermentation, okay, is another way. But that we do want to read about. So anaerobic respiration. Some cells can function for a short time when oxygen levels are low. Does anyone remember why we had said, where is oxygen going first? The first priority is where? To the brain. Then another, now in this case it would be a muscle. Okay, so this needs oxygen. Your diaphragm is a muscle. That needs oxygen. And then, okay, what are probably the largest muscles in the body, do you think? And there's a group of 3 on the back side of your leg, and there's a group of 4 on the front side of your leg. You have your quadriceps, and then your hamstrings, which is your, and of course those are long drug-out names, but we will talk about them in the spring. But those muscles are quite large, and they require large amounts of oxygen. Okay, and we're going to explain why that's important here in just a moment. Okay. Some prokaryotes are anaerobic organisms. They grow and reproduce without oxygen. In some cases, these cells continue to produce ATP through glycolysis. No surprise, that's why we put that up here. However, there are problems with solely relying on glycolysis for energy. Okay? Even if we didn't go any further, if we're talking about glycolysis only, why would that present a problem for producing energy? It has something to do with down here. Am I hitting the right spot? Nope. How efficient is this? Not very. How efficient is this? Well, what's going to give you more energy? This side or this side? Certainly this side is going to give you a lot more energy. Okay? So, but sometimes this has to take place because your brain, your heart, your diaphragm, those also always have to have continual supply amounts and supplies of oxygen. Okay, so glycolysis provides only 2 net ATP for each molecule of glucose, and the cell has a limited amount of NADH. Okay? Glycolysis will stop when all the NAD plus is used up if there is not a process to replenish the NAD plus. The anaerobic pathway that follows glycolysis is anaerobic respiration or fermentation. Fermentation occurs in the cytoplasm and regenerates the cell's supply of this NAD plus while producing a small amount of ATP. The 2 main types of fermentation are lactic acid fermentation and alcoholic fermentation. Which one do you think we're going to talk about? Well, we might think that that would be ideal, but no, we're not going to talk about alcoholic fermentation, rather lactic acid. Okay, so on the next page on 232, okay, Lactic acid fermentation. In lactic acid fermentation, enzymes convert the pyruvate made during glycolysis to lactic acid as shown. This involves the transfer of high-energy electrons and protons from NADH. Skeletal muscles produce lactic acid when the body cannot supply enough oxygen, such as during periods of strenuous exercise. When lactic acid builds up in muscle cells, muscles become fatigued and might feel sore. Lactic acid is also produced by several microorganisms that often are used to produce many foods, including cheese, yogurt, and sour cream. So, to wrap this up, and where we'll start tomorrow, okay, when we take both of these and put them together, now you don't need to write this down because we're going to go over it again at that time. But notice, for every molecule of glucose, okay, what's going to happen is it's going to go through this process we called glycolysis. Okay. Then it's going to try to enter the Krebs cycle. Okay. But in order to enter this cycle, something must be present to keep on going. Okay? Something must be present over here to get into this Krebs cycle. You have to have oxygen. So this is one requirement, oxygen. Okay? And then there's another requirement, and that's called acetyl coenzyme A. Okay? So if we back up just one page to 230, okay, figure 13 is talking about this entire process. Okay? Notice that it says pyruvate from glycolysis. Okay? So what it's talking about here, this is what we're referring to, this diagram right here. Okay? It says pyruvate from glycolysis. Okay? How many ATP are we getting from this glycolysis? Now the net, not the overall. How many do we get from that? Just this, just only 2. Okay? Now, when you look a little further down, it says acetyl coenzyme A. So that is required for this to enter the Krebs cycle. So now, we've talked about 3 enzymes in this chapter. Do we know what they are by chance? Because you have to include the Calvin cycle. So what enzymes have we talked about? Ribisco is one. What's another enzyme we've talked about? I said I was mispronouncing it the whole time. And you can find that in figure 14 on page 231. In the bottom right-hand corner of figure 14 on page 241, there's another enzyme. It's called this. ATP something. Synthase. Synthase. Yes. See, I was always pronouncing it as synthetase. Okay? And then, what is our third enzyme? We just introduced it 2 minutes ago. We know it's an enzyme because it's in the title. It's right here. This is the third enzyme, acetyl coenzyme A. Those are the 3 main enzymes that we've talked about in this chapter. Okay, so where does that put us? A good spot to talk about photosynthesis and cellular respiration on page 233, doing that tomorrow. Okay, you have some papers that were assigned last week. We will collect them tomorrow. So you have just a few more minutes. We have talked about enough content now that hopefully we could wrap those papers up. Okay, so that's all I have. We'll catch up to you next time.